everybody. I've got a couple videos coming up to talk about our first section of Unit 2. This would be something that would be very good for you guys to be ready for and know what you're talking about on our first day back to school, as this is going to be something that I'm going to go over in class, but quickly, because we've lost so much time with the delay here the last couple of weeks. So a big thing that's different about Unit 2 than a lot of the Unit 1 material is that it kind of builds on itself. So the material that we cover now is going to be really important for Subunit 2. And these being your first grades of the third grading period, doing well on this one's going to be important. So directly following this video, we're going to just segue directly into going into the notes. I'm going to make some drawings and show you guys a couple things, and there will probably be one or two additional videos posted after this video. As always, if you guys have any questions with what you see or what I do on these videos today, feel free to email me. And like I said, we're not going to spend a great deal of time on this in class, so you're expected to kind of know what we're talking about on your first day back. So here we go with the first part of the next video. Okay, so here's the notes. You guys should have seen these online or printed them off or whatever. Now I'm not going to read you the whole notes packet, but I do have some things circled and highlighted as we go through. If you want to go through and use that as kind of a word bank sort of thing of words that you really need to make sure that you know, it'll also be related to the homework for this section. You can do that, and again, you have the ability to just pause this video, because I probably won't stay on a lot of these pages very long. Like I said, I'm not going to read it to you, so you can just pause the video and get those words. So obviously nothing highlighted here, you guys can read that, and just because we're skipping things doesn't mean it doesn't have value. It's just not maybe a vocab word that's going to end up on your quiz. You guys learned a long time ago that DNA stands for deoxyribonucleic acid. Per usual, if we use an abbreviation, I'm going to want you guys to know what it means, so that'd be something good to know. Definition for what a gene is, that's going to be valuable. Again, I'm not going to read it to you right now, I'm just kind of drawing your attention to some basic things. What alleles are is different types of genes. Locus, another vocab word that we'll use. Genome. And again, you guys should be reading through this packet yourselves. See, nothing highlighted here doesn't mean you shouldn't read it, just not something I was going to make a point of. Definitions for protein, amino acid, and nucleotide. This one's going to be big. We'll talk about in a second. So, talking about those parts of a nucleotide, and this is going to be the first drawing we're going to look at here. Those parts are a 5-carbon ribose sugar, which in DNA is called deoxyribose. A phosphate group, which is a phosphorus atom that's surrounded by oxygen atoms. I don't necessarily care that you know that, but if you wondered, that's what it is. And then one of 4 nitrogen bases, which in DNA are adenine, thymine, guanine, and cytosine, and I think that those words should be familiar to you. If you've heard of uracil before, that is used in RNA, and it replaces thymine. So where thymine would be in DNA, in RNA it'd be uracil. So my first drawing here, if we start over here, so this is just a good old DNA double helix. I imagine that everybody should be familiar with that. And then we have my classic zoom in box. And so I zoomed in and I drew with a little bit more detail. So the 5-carbon sugars are these little pentagram looking doodads. And then my phosphate groups are these little circles. And they are attached by a type of bond called a phosphodiester bond. It's a type of covalent bond. This bond is very strong and shouldn't break during that organism's lifetime, or for that matter, for some time after. If any of you have heard about where they're trying to gene sequence things like woolly mammoths and things from like 10,000 years ago, in many cases those bonds still haven't broken when that animal's been dead for thousands of years. Granted, it's been frozen, but still. So it's a very strong type of bond. That bond will not break. And then the other part of our DNA are these nitrogen bases. I've got A's for adenine, T's for thymine, C's for cytosine, G's for guanine, and if you look at all of them, you'll notice that you always have a thymine with an adenine, or an adenine with a thymine, and a cytosine with a guanine, and a guanine with a cytosine. So those are what we call complementary base pairs. And I kind of color coded over here too. Again, you guys could pause this if you wanted to look at it longer. And then if we look down here, so this is what we were looking at in the notes with the parts of a nucleotide slide. So this is a nucleotide. And if you guys look back up here, I kind of drew a circle around this one and then just made it bigger. So that is my phosphate group. That is one deoxyribose, or a 5-carbon sugar, and that's one of those nitrogen bases. So this is what a nucleotide is. So DNA, in turn, then, is made up of a bunch of nucleotides. There's one, there's one, there's one, all the way, up and down the entire double helix. So that was something that I wanted to look at, show you guys some pictures of. Going back and looking at the notes, double helix, we already talked about, pretty basic vocab term, but it's still good to know that. We just mentioned all these things. So structures of DNA, we talked about deoxyribose being that 5-carbon sugar. We talked about phosphate groups. We talked about phosphodiester bonds. We talked about the nitrogen bases. I didn't mention that the nitrogen bases are held together in the center by hydrogen bonds. So these bonds are much more easily broken. So these bonds are designed to break, which you'll see in the last video, where these phosphodiester bonds do not break. It's by design that those bonds are able to be broken, and you'll see why in a bit. This picture looks probably a lot like my picture that I drew. If you look on the online version that I sent out, it's going to look a lot better than this, but you guys should be able to see a little bit with the 2-ring and 1-ring structures if you look at that, not on this video. We talked about which ones were complementary base pairs. Adenine and guanine have this double ring structure I'll show you in a second on the next page. Those are called purines. That's just the name of them. Again, if you don't like it, I didn't name it, so I don't know what to tell you. And then the other two, thymine and cytosine, are called pyrimidines, and they have a 1-ring or single ring structure. And to look at a picture for that, so this one's adenine, this one's guanine, and what it means by double ring is that this organic chemistry model has 1-2 rings, where if we look up here with the pyrimidines, thymine and cytosine only have 1 ring of atoms. So that's what that means. And they're designed to fit a purine with a pyrimidine. So those two would go together. And if you look, again, at the slideshow, that's a 2, that's a 1, that's a 1, that's a 2. And if you can't really tell in this picture, it's just because it's a printed version and it's not that great. Introns and exons, this will be important for stuff we talk about later. So make sure you know the difference. This is one of those classic biology moments where it's 2 words that sound pretty similar but mean almost opposite things. So make sure you know which one's which. And the introns are sometimes called, like I said up here, junk DNA or non-coding DNA or intergenic DNA. So it's just different ways of saying introns. We'll talk about that more later. Some chromosome information, which we talked about at the end of Unit 1, but we're going to revisit now. We talked about sex chromosomes briefly. If you're a male, you have an X and a Y chromosome. If you're a female, that 23rd pair is 2 X chromosomes, and we'll talk about that again more later. We talked about chromatids, centromeres, and chromatin last time, so this is kind of review, but hopefully you guys know what those words mean. And this takes us into my second drawing. So we're going to go back over here. And so this is drawing 2. So again, feel free to pause so you can look at this longer than the video is holding it. So this is a homologous pair of chromosomes. So there are 2 chromosomes for the same pair. This is stuff that we learned last time. So this is 1 chromosome. The 2 halves are the chromatids, so divided by this line. So this is a chromatid and this is a chromatid. They're held together in that constricted area in the center called the centromere. So all that was old news from last quiz. The new thing with this, though, so the smaller arms are called the P arms. Notice that these ones are shorter than these ones, and then the longer arms are called the Q arms. The reason for the name is for gene naming, which we're not going to get into, but it makes it more easily known where a certain gene is located for scientists that are actually using that information. But so the short ones are the P arms, the long ones are the Q arms, and that's the case for any chromosome. And then the other new word was histones in the notes. So these are specialized proteins that the DNA wraps around to form the chromosome shape. So I tried to draw this, which again, it's the best as I could do. So the histones are all my little black dots in there. And then so my DNA is the red squiggle. And you can think that the DNA is using those histones as a thing to wrap around to form that chromosome shape. This is not the best thing I'll ever draw you, but I think it gets the point across. I'm going to stop this video here so that it's not terribly long, so it doesn't take me forever to upload it. But we should have one more video upcoming, and the next video is going to be the big deal for this set of notes. So we'll see you then.